certain plants. For example, this tomato, if we want to increase its shelf life, particularly whenever the tomato gets ripened on a plant, its shelf life will decrease. That is, it can stay only for up till 2 to 3 days. After that, it starts becoming rotten. So to increase the shelf life of this tomato plant, we can modify its genome such that the shelf life of this tomato increases, such that there will be less loss of this crop, and in turn most of the productivity will be useful for serving as a food. So for increasing this shelf life, we are modifying the genome of tomato plant by genetic engineering techniques. Why it is genetic engineering is we are engineering genes. Engineering is modifying the actual mechanism is known as engineering. So we are using genetics to engineer these plants. So hence it is known as genetic engineering technique. Here we add a gene to increase the shelf life, for example, or if there is any detrimental gene, any gene which is causing loss to this plant, we can remove that gene. For example, some toxic genes will be present in some plants. So that can be removed by genetic engineering techniques. This is known as transgenics. That is, we are modifying the genomes either by introducing some genes or by removing some detrimental genes. So this tomato here, it is gene silenced, means the genes which are responsible for early ripening of tomato is silenced using RNA interference technique. So here, with the help of RNA interference technique, the gene which is responsible for decreasing the shelf life, that is because tomatoes get ripened with the help of an hormone known as ethylene. So if this ethylene production is shut down, then the tomatoes will not get ripened soon. Easily we can increase the shelf life, and whenever we need, we can spray the ethylene to ripen them. With that, this if you see this ordinary tomato, in 20 days it will get rotten. It begins to rotten, and in 45 days completely it spoils. Whereas the gene silenced tomato, it will be fresh for up till 45 days. So this is the transgenic technology with which we can improve many qualities of our plants. Not only increasing the shelf life, there are many other qualities or applications of transgenics which can be improved using genetic engineering technology. Then before that, we will see some crop modification techniques. Apart from transgenics, we have different other modification techniques like crossbreeding, mutagenesis, polyploidy, protoplast fusion, transgenesis, and genome editing. Coming to crossbreeding, this is the age-old process. From thousands of years, it is carrying on, where two sexually compatible species are crossbreeded to obtain the desired traits. Means this two organisms or plants of same species or compatible species are crossed with each other. Sexual crossing is done here to create a new variety with desired traits. This is an age-old process where we can produce hybrids. Coming to mutagenesis, here we introduce certain mutagens to improve the qualities. So use of mutagens such as radioactivity to induce random mutations, creating the desired trait. Here, next one is polyploidy, where we can multiply the number of chromosomes and which create certain important characteristic like seedless watermelons. Seedless watermelons are created by crossing a plant with 2 sets of chromosomes with another that has 4 sets. The seedless fruit has 3 sets. So if you cross 2 watermelons, one with diploid nature and one with polyploid nature, that is 4 sets of chromosomes, you will get a seedless fruit variety where you have 3 sets of chromosomes. In this variety of watermelon, where you see 3 sets, there will be no seed, or it is known as the famous seedless variety. So that is, we are improving a trait here. Trait is nothing but a character, like improving the color of a fruit, removing the undesired characteristics like seeds of watermelons, which may disturb while you eat them. So removing these undesired characters or adding any desired character, like improving the color of roses and creating different varieties of rose. These are known as traits or characters. So with the help of these techniques, modification techniques, you can improve the traits. Means get the desired trait. The next one is protoplast fusion. When you fuse two cells without any cytoplasm and without any cellular membrane, and only protoplasm is fused, then desired traits will be obtained. Like here, a trait known as male sterility is transferred from radishes to red cabbage by fusing their cells. Male sterility helps plant breeders make hybrid crops. So this is one of the technique, male sterility, which gives some desired traits in crops. That can be done using a technique known as protoplast fusion. Then comes our technique known as transgenesis. So transgenesis is addition of genes from any species to create a new variety. So we are altering the genome of an organism by either adding the gene or by removing a gene. So this addition of genes from any species to create a new variety, because we are using genes from other varieties, it is known as transgenesis. If you use genes from the same species and transfer into plants, in between plants of same species, it is known as cisgenesis. There you do not have any regulatory bodies, or there will be no control, because from the same species you are transferring different traits. Here, because we are using different species to create a new variety, like we are using papaya and we are using a bacteriophage here. So this is a plant and this is a bacterium microorganism. So as we are using different species, it is known as transgenesis. Genes are added from different species to create desired traits. So here, this papaya, it has some ring spot virus. This virus will damage the productivity of papaya crop, leading to huge loss. That ring spot virus resistance can be brought in this papaya using transgene technologies. This papaya, which is resistant to virus, is known as rainbow papaya. Rainbow papaya is modified with a gene that gives it resistance to the papaya ring spot virus. So when you introduce a gene, outside gene into papaya, it is known as transgenic, and this transgenic will become resistant to ring spot virus. In turn, you will get large productivity without any huge loss in the production. Then comes the another technique. This is the current trend which is running, known as genome editing, where you edit the genome using an enzyme system. This we will discuss, but the mainly we will focus today on transgenic plants. So these are the we are saying that in transgenesis we are modifying the genomes either by adding a gene or by removing a gene. So for adding or removing these genes, we require some techniques known as gene transfer techniques, because we are transferring a gene from one species to another species. So these gene transfer techniques are of two types. Either you can directly transfer a gene from one species to another, or you can indirectly, another method is indirect method, where indirectly you can transfer the genes from genes in between different organisms. Coming to direct methods, the most important direct method is known as biolistic method. This is also known as particle gun method or gene gun method. Here you use a gun like our gun system. Here the particles, heavy metal particles like lead or gold particles, metal particles, they are combined with our gene of interest, and they are shooted into the plant cell. That is known as biolistic method or gene gun method, because here directly we are introducing the gene into a plant cell using some heavy metals. So here the heavy metal, mostly the gold particles are used. The gold particles are combined with our gene of interest, and these particles are directly inserted into the cells of plants using gene gun method. That is known as direct method of gene transfer. Coming to indirect method, these are all different direct methods. The most often used method is biolistic or particle gun method. Then we have polyethylene glycol method. Then we have like silicon carbide method, micro injection, macro injection, ultrasonication, and electroporation. Then today in our class we will focus on this indirect method known as agrobacterium tumefaciens or agrobacterium rhizogenes method. This agrobacterium tumefaciens is a bacterium that causes crown gall diseases in plants. That is, it is a tumor causing disease. Means whenever a wound is seen in a plant, this bacterium will enter into that plant and it causes tumors in that plant. This is the negative effect of this agrobacterium tumefaciens. But the genetic engineers, they used this negative effect of agrobacterium and created a positive response, that is but as this agrobacterium tumefaciens is uh, uh, has the capacity to get inserted into plant cells, this this agrobacterium is used in a positive way to introduce certain genes. Some important genes are introduced into the plants through this agrobacterium tumefaciens method. So
Madam, there
let
are
us
some
see.
annotations. Is there any difficulty from participant side? There are lines coming on the screen. Is it from somebody, students? Hello? Javriya
Maybe,
madam.
sir. Maybe, sir. I'm not doing that. Some other ones
Okay,
are
okay, okay.
doing
Wait,
that.
wait, wait. Let me suspend the activities 1 second.
Sir.
Now you start sharing the screen again from there itself. Yeah, now you can start. Now there will be no annotations. You can unmute yourself, Javriya madam. Yeah, you can start your video also because you're co-host. You can do that. Yeah,
Okay.
I did not log the meeting today.
So
That's
today,
not annotations. Now I saw annotations, so I stopped it. Sorry for the
okay.
inconvenience. You continue.
Yes, sir. No problem. So as in transgenic, we transfer different genes. This gene transfer can be done by two methods: direct method or indirect method. In direct method, directly you can introduce the gene into a plant cell, whereas in indirect method, you can use an mediator. That mediator will be either mediator will be a bacterium, that is agrobacterium tumefaciens. So this is the way of creating transgenic plants using agrobacterium tumefaciens. So this is an agrobacterium. It is a bacteria which has, apart from its own chromosome, it has a plasmid DNA. Plasmid DNA is an extracellular circular genetic material which has the capacity to transfer. So this TI plasmid has an area known as TDNA. This TI plasmid has an area known as tDNA, transfer DNA. In this transfer DNA, you have restriction cleavage site. So here you can break the tDNA and introduce your gene of interest. The gene, for example, any gene which can be taken from another species, this is known as foreign DNA. This foreign DNA can be inserted into your TI plasmid. So now when this foreign DNA is introduced into the TI plasmid, it becomes a recombinant plasmid. So the main criteria for introducing this foreign DNA is you should have promoter and terminator regions here. So if you have a promoter and terminator regions, the gene can be easily transferred in between generations. So this TI plasmid, this part of known as tDNA or transfer DNA is non-essential. So if you remove this part also, there will be no problem for inheritance of this TI plasmid, because these are the essential genes. We should not disturb them, and only the non-essential part of TI plasmid, there we have to insert our foreign DNA or foreign gene. After inserting this foreign DNA, this plasmid, that is TI plasmid, which has tDNA segment, it is now known as recombinant TI plasmid. This recombinant TI plasmid, after introducing our foreign gene or gene of interest, is again reinserted into the bacterium. So this, from this agrobacterium tumefaciens, first you have removed this TI plasmid. After removing by different genetic engineering techniques, then you will insert your foreign DNA. After inserting the foreign DNA, now it is known as recombinant TI plasmid. It is reinserted back into the bacterium. After reinserting, it is co-cultivated with your host plant. The bacterium is used to insert the tDNA carrying the foreign gene into the chromosome of the plant cell. Okay, now this TI plasmid, it has our gene of interest. This gene of interest is inserted into the plant cell by doing co-cultivation. Means your plant cells and bacterium are grown together. Uh, uh, Now, due to a technique known as transformation, these genes of interest will be inserted into your plant cells. Now you will get your plant that a plant is generated from a cell clone. All of its cell carry the foreign gene and may express it as a new trait. So when all of your plant cells have taken this agrobacterium tumefaciens into it, because this we have studied that this has the property to infect plant cells to cause crown gall diseases. That property we are taking and using it in a positive way. So as this agrobacterium has the property to infect plant cells, here it will infect the plant cells. When it is infecting, this agrobacterium gets inserted into this plant cell. When agrobacterium is getting inserted into the plant cell, along with this, our foreign gene, that is gene of interest, is also getting inserted. When this gets inserted into the plant cell, it will give new trait. So this is a very complicated mechanism. So because we are discussing on applications, I'm not going into detail. So general, this bacteria gets inserted into the plant cell, and now when you grow this plant, it will show you the desired trait or desired character. This is the agrobacterium tumefaciens mediated transgenics. So this is the first transgenic plant which was developed. That is a tobacco plant which is resistant to antibiotics. Because here this is the sensitive plant. So this is the resistant tobacco plant. This is the first transgenic plant which has a gene known as NPT2 gene. This is neomycin phosphotransferase 2 gene, which gives resistant to different antibiotics to the plant. So once you insert this gene into the tobacco, this gene is taken from bacteria. From bacteria, this gene is taken and it is introduced into tobacco plant. So there is transgenics between bacteria and tobacco plant. From bacterial, a gene is taken, a desired gene is taken, and it is introduced into a plant, such that now it will become antibiotic resistance. No bacteria can attack this plant. So this one is the first transgenic plant developed. After that, this stream flourished very fastly, and now we have number of transgenic varieties which are in the market. Since then, with the rapid development in plant molecular biology and genetic engineering technique, a wide variety of transgenic plants with important agronomic traits such as pest resistant, drought tolerance have been developed. Now we see many varieties of transgenic plants which are developed, which show us important traits. So these are the different traits which are present in plants. Like you can by creating transgenic plants, you can improve the nutritional quality. The nutrition of your plants may be improved by uh, introducing genes. Then you can develop hybrids for giving higher yield. This is the main issue. So when you have high yield, the productivity will increase, and that will help in serving food for this increasing population. Because there is an urgent need of food security, because the population is increasing day by day and we have to provide food to all the increasing population. In this case, transgenic plants will help us in creating hybrids which give higher yield. Then we can introduce biotic stress tolerance in transgenic plants. Biotic stress tolerance is the stress caused due to living organisms is known as biotic stress. Many living organisms like bacteria, fungi, viruses, then parasites, different protozoans, they cause diseases in plants. And as these disease plants give less crop yield, productivity will decrease due to biotic stress. Because when the plant is in stress, automatically the productivity will decrease. So by introducing transgenic plants, we can make plants biotic stress tolerant. Means no insect or no pest or no virus can attack the plant if you introduce certain genes into the plant. So automatically the plants will become healthy and the crop productivity or yield will increase. Same way you can make the plants abiotic stress tolerant. Abiotic stress is stress caused due to environmental factors. Biotic is stress caused due to living organisms like pest, insect, viruses, bacteria, etc. Abiotic stress is the stress caused due to environmental factors. Like if you have floods, more water reaches to plants, that will disturb the metabolism of plants. Then if you have drought, then there will be no water, due to which the plant mechanism get disturbs. When plant get disturbs due to drought, there will be no water availability to plants, and hence plants will get dry and ultimately causing death. So that will be avoided if you introduce certain genes which are drought tolerant. Then you have salinity. Many soils, due to modern techniques or due to use of fertilizers and pesticides, they are becoming saline or alkaline. Means more salt is getting accumulated into soils. This saline soil will make soils less productive. The plants which grow in this soil becomes less productive due to uh, salinity or alkalinity. Soil may become either acid or base or saline, or the pH of the soil gets disturbed. Due to that, plants do not grow properly. So if you introduce certain genes which can resist salinity also, then your plant become stress tolerant. So automatically when it becomes stress tolerant, it will become healthy and productivity will increase. The root cause of all this is to increase the productivity by removing or by reducing the diseases, by removing undesired characters in the plants, you will increase the productivity. That is the main cause of transgenic plants. Then with the help of transgenic plants, you can also produce pharmaceuticals and edible vaccines. Like vaccines you can produce using plants. This will reduce the cost of manufacturing of vaccines, because now vaccines are manufactured in huge bioreactors in factories and industries that will cost you more amount. And with the help of transgenic plants, you can farm grow your vaccines in farms directly. You can produce vaccines. So then other pharmaceutical medicinally important products also you can produce using transgenic plants. Like and one more important trait is you can enhance the shelf life of plants using transgenic technology. These are the different applications of transgenic plants. So one by one we'll see the different applications. First one is herbicide resistance. So here in herbicide resistance, this is one of the trait introduced using transgenic technologies, where in this picture, if you see, there are many weeds growing, unwanted plants growing in between fields. Here you can see transgenic plants where there are no weed growth. Means unwanted plants, weeds which are not useful are avoided here. Whereas here, if you see along with this crop, you have all the unwanted or undesired plants which are of no use. So here, with the help of with the help of transgenic technologies, you can make plant herbicide resistant. Means here, if you spray herbicides also, herbicide is a medicine, or it is a drug which is used to kill herbs or kill weeds. So they will kill unwanted weeds. When you spray these herbicides here, these weeds will get killed, but along with that, our crop also will get affected. So if you make this crop herbicide resistant, even though if you spray your herbicide here, the chemical which you use to kill plants, if you spray here, these unwanted plants will get killed. But along with that plants, our own plant also gets killed. But if you create herbicide resistant varieties and if you spray the herbicide here, the unwanted weeds will get killed, but your plant will not show any effect. It will grow healthily because here it has a gene which is herbicide resistant. Because of that, it will not show any effect even if you spray any amount of herbicide here, right? So genes for resistance to certain herbicides have been introduced into crop plants so that they can thrive even when exposed to herbicides that kill weeds. So when you expose these plants to herbicides also, they will not get killed, but only the unwanted plants they get killed. This is the one of the most important technique application of transgenics, where you can introduce genes which give herbicide resistance. So these are the two plants. A coffee plant, this one is herbicide resistant and this is herbicide sensitive. So if you spray herbicides here, it will not show any effect. Whereas this plant, because it is not transgenic one, there is no gene for giving resistance to herbicide, so it will get disease attacked when you spray herbicide here. So this is the pathway which is attacked in creating herbicide resistance gene. So actually there will be a chemical known as glyphosate. This chemical glyphosate is present in herbicides. This glyphosate will block this pathway. This is an amino acid pathway where three important aromatic amino acids are generated, are synthesized in plants for their growth. These three amino acids are aromatic amino acids which are very important for plant growth and metabolism. So when you spray herbicides, that herbicides, they contain a chemical known as glyphosate. This glyphosate will block your pathway here. This is the pathway where shikimic acid is synthesizing three important amino acids. In all plants, this pathway will be present, either in your weeds or in your crop of interest. In all plants, this pathway will be present, and with this pathway, amino acids will be synthesized, which help in growth of plant. When you spray herbicide, this glyphosate is present in that herbicide, which will block this pathway in this step. So this EPSP synthase enzyme will be blocked due to this glyphosate. So when this enzyme is blocked, this substrate will not be converted into this product. When this substrate is not converted into the product, three amino acids will not be synthesized. When these amino acids are not synthesized, this pathway will get stopped or blocked, and the plant will not show normal growth, and the cells gradually they die because of absence of these essential amino acids. So this glyphosate is blocking here. Now in resistant genes, we are blocking this glyphosate, we are making this glyphosate inactive here, so that this pathway continues in our crop of interest. In these plants, this pathway will be active, whereas in these weeds, because there is no resistant gene, this pathway will get blocked with the by this glyphosate. So only your glyphosate resistant genes will survive, the plants will grow, others will die. So these are the this is Roundup Ready spray, which is uh, herbicide. This Roundup Ready is the most famous spray available in market to spray on plants to get rid of these weeds. These are the weeds which will be get ridden with the help of this spray, which contains glyphosate. So this glyphosate present in Roundup Ready spray, it will block the shikimic acid pathway in these weeds. But because these plants are resistant, the pathway will not be blocked here. Automatically the weeds will, all the weeds will die, and your plant will grow healthily. One of the disadvantage here is slowly these weeds become herbicide resistant. So after some years, this work is actually in progress now. To avoid herbicide resistance, genetic engineering works are going on. So there will be, if herbicide resistance comes into these weeds, then it will be very difficult to manage them, right? So this is the about herbicide resistance, where this pathway is blocked by herbicides in sensitive plants, whereas in resistant plants this pathway is not blocked, creating essential amino acids. Then coming to the next application of transgenic technologies, it is insect resistance. So many plants, for example, in our areas we see maize and cotton, they get affected by many caterpillars. These are the insects which affect cotton plants and maize also. These insects, they will directly attack the fruit of cotton, such that the cotton quality which we get will be very deteriorated quality. Undesired quality of cotton will be obtained if this insect feeds on the, this is known as cotton bollworm insect. This is the caterpillar of that insect. So this insect, it goes and feeds on the cotton fruits. When it feeds on the cotton fruits, the quality of cotton fruit will get deteriorated. So the cotton which we obtain will be of very weak quality. So here what we are doing is, with the help of transgenics, we are introducing genes which make these fruits insect resistant. So whenever this caterpillar goes and feeds on the fruit of cotton, because of the resistant gene present here, the metabolism of this insect gets damaged. Whenever it feeds on this fruit, the protein which is present in this, the gene which synthesizes protein, this protein will damage the gut or intestine system of this insect, and that insect will die. So that is known as anti-insect strategy, where we are not using any insecticide, but the plant itself is creating its own insecticide. See, whenever you plant cotton plants, uh, we use insecticides that are harsh chemicals to avoid insects, to avoid any pest to attack the plant. So these insecticides may affect our healthy plants also, or it may affect our soil also. So that will be undesirable. So what we are doing here is, without any spraying of insecticide, we are protecting the plant from insects. How this plant itself, it is acting, it is producing its own insecticide with the help of transgenic technologies. So here in these cotton plants, we are introducing a gene from a bacterium known as Bacillus thuringiensis, BT. BT from a bacterium known as Bacillus thuringiensis, we are introducing a gene known as cry gene. Cry means crystal. This is a toxic gene. So when this gene undergoes transcription and translation in cotton plant, after introducing, when you introduce this bacterial gene into cotton plant, it will undergo transcription translation to synthesize a protein known as crystal protein. We are not introducing cry protein, but we are introducing cry gene. From that cry gene, you will get a protein known as cry protein, crystal protein. This crystal protein is toxic to insects. So whenever this toxic crystal protein is synthesized in this fruit, and when insect attacks it, caterpillar of that insect attacks, this cry protein will enter into the metabolism of this insect, to the gut of this insect. When it enters, it will damage the gut of this insect, and this insect will die. So this plant is automatically creating its own insecticide. Okay, this type of insect resistance was introduced in corn, in cotton, in tobacco, and in other plant species. So these use cry gene, which is present in Bacillus thuringiensis. This cry gene is inserted into plants, which produces a protein that causes paralysis and death of many insects. So this is the mechanism. Bacillus thuringiensis is a bacterium which is found in soil. In this bacterium, Bt bacterium, we have a protein. That protein paralyzes the insect's digestive system. So the insect dies of starvation. The gene responsible for coding this protein is inserted into the DNA of plants. Actually, this protein is present in bacterium. This DNA from this bacterium is inserted into the plant. Now this plant, when gene is inserted, it will synthesize its protein. When this protein, the plant then produces the protein that is responsible for killing insects. So its own personal insecticide. So Bacillus thuringiensis, we are taking the Bt gene, and we are introducing into the plant. The pest dies when feeding on any plant part, because in all the plant cells this gene will be expressed. This is the insect resistant technology, which is helping most of the crop plants in the world. Then gene in crystal toxin, okay, gene for Bt toxin was transferred to 10 Bt transgenics. Is very, they have Bt corn also, Bt brinjal also. All these have this cry protein, cry gene inserted into their genomes. This cry gene, it synthesizes inactive protoxin. Then proteinase digestion in insect gut makes the active toxin. In the plants, this toxin will be inactive. But when insect consumes this plant, the digestion insect makes this toxin active.
Madam, your sound is not audible.
So inactive.
Your share screen is gone, ma'am. Yes, okay. You unmute yourself, Jabriya ma'am.
Okay, sir. So this cry gene, it will be in inactive stage in plants. But when insect consumes this, eats this plant, the inactive gene toxin becomes active because of the metabolism of insect. After becoming active, it will go and attach to the cellular membrane of insects. To all the cell membranes, it will go and attach. After it attacks on the cell membrane, it creates a pore. Sir? Sir, is it okay?
Yeah, okay. Sorry. It is my mistake.
Sir. So this protein goes and attaches to the cell membrane of insects. To all the cell membranes, it will go and attach. It will create a pore here. Through this pore, all the electrolytes of cells, they leak. Whatever the electrolytes they are present in the cells inside the cytoplasm of cell, they will leak, ultimately causing the death of insects. So because this toxin is inactive in plants, it will not cause any damage, any harm to the plant, but it will harm only the insect. That's why bt cotton is not at all causing any damage to the plant ecosystem. It is only causing damage to the insect which is attacking it. So sir, I'm getting this uh, slide here which is showing please move this window away from the shade application.
No, this is from your side only because it is not it is from our side. From long time it is appearing there. Yeah, now gone.
Okay, sir. So we have seen two characteristics of transgenic plants. One is herbicide resistance, another one is insect resistance, then third one is virus resistance. Again, it is appearing here, sir. What should I do?
It is not our side, actually it is happening from your system itself because annotation can be done by participants. This can't be done by participants actually.
Okay.
It is okay now.
Yes, it is appearing and it is moving again. Okay, sir, no problem. Let it be. So here, virus resistance. The third characteristics is virus resistant. This is the papaya. This papaya, once it affected with ring spot virus, that virus caused a devastated loss to the papaya industry. In Hawaii, there were many papaya growers whose all fields got damaged due to this ring spot virus. Then we have using transgenics, virus resistant papaya was introduced, which saved their uh, industry, because in this a gene from the virus which encodes a protein in the virus outer coat is copied into the plant's DNA. Viral protein code was copied into the plant's DNA. The plants then makes the code protein which is harmless but which stimulates the plant's natural defenses. It will act as providing immunity to the plant. So when the viral code protein, because in viruses mostly the genetic material will be harmful, means it will be responsible for causing any infections or diseases, but the protein code will be harmless. Most of the viruses, the DNA or RNA containing in them will be causing diseases, whereas the protein coat will be harmless. So here in papaya, the protein coat of viruses was injected. When you inject the protein coat, it will not do any harm to the plant, but it will stimulate the plant defense. The plant defense thinks that virus has infected me, and it will stimulate its immune system, means it will activate its immune system, creates antibodies or creates any T cell mediated immunity. So the immune system of papaya plant becomes strong. But as there is no harmful item, it will be present in as a memory in the papaya plant. Wherever any virus it infects this papaya plant, the immune system which is present as a memory, it will attack that virus and it will kill it. So what we are doing here is we are injecting a viral gene into the genome of papaya plant. That viral gene has only harmless thing, that is protein of virus. DNA or RNA of virus is not present, only protein is present. That protein will be thought as a diseased agent by the papaya plant, and it will stimulate its immune response. When immune response is stimulated, that will stay as memory in the papaya plant. This memory will be again activated when actual virus comes and attacks. So whenever there is any viral attack, this memory will stimulate in the plant and it will kill that virus. This is the virus resistant papaya created. Now in, if you see in, this is the rainbow papaya, which was genetically modified by Dennis Gonzalez. This was introduced in 1992, and it saved all the papaya industries because that virus was very devastating and it destroyed all the naturally wild papaya plants in the world. So now you have only the hybrid varieties of papaya present. Uh, so here, this is the one of the major breakthroughs in the transgenic technology, growth of rainbow papaya, which is virus resistant. So this technology was applied to many other plants after introducing in papaya. This is one of the important trait of transgenics, important applications of transgenic, that is virus resistance. So we have discussed about herbicide resistant, insect resistant, and virus resistant. Then the third one, another application of transgenic technology is improving shelf life. Shelf life of fruits and vegetables can be improved using transgenic technology. So here you can see tomato where you can delay the fruit ripening, because tomatoes, when you pluck it in the farm and when you or in the field, and the time when you bring it to the market, half of the papaya crop will be spoiled, because in papaya there will be lots of ethylene production which will help in ripening of fruits. So when fruit gets ripened, it will easily damage due to, and due to, because this fruit is very soft, it will get damaged during transport also. So here if you delay the fruit ripening in tomatoes, then unripe fruits can be brought into the market. Then we can increase the shelf life also. Normal our wild tomatoes we can save only for up to 2 to 3 days, or most probably for up to 1 week. But this type of tomatoes we can store them up till 1 month without any disturbance in their metabolism. So here we use a technology known as antisense RNA technology. So RNA, as you all know, RNA is single stranded, and it will function in a single stranded mode. But when you make this RNA double stranded, it will not show its function. Okay, so we use a technology known as antisense RNA technology, where the single stranded RNA of our plant's metabolism will uh, bind with this antisense RNA, and thereby making the RNA inactive. Okay, the RNA which is active in living organism is the sense RNA. To this sense RNA, if you make it double stranded by adding the antisense RNA, the function of that RNA will stop. So here this antisense RNA technology was used to delay fruit ripening. Here some plants like tomatoes were used in this technology to increase the shelf life. So most tomatoes that have to be shipped to market are harvested before they are ripe. Otherwise, ethylene synthesized by the tomatoes causes them to ripen and spoil before they reach the customer. Ethylene synthesis is responsible for ripening of tomatoes and spoiling of tomatoes before they reach the customer. So what we do here is we will construct an antisense RNA that will be complementary to the mRNA for an enzyme involved in ethylene production. So for ethylene production, the mRNA responsible will be blocked or silenced by making this RNA combine with another antisense RNA. This antisense RNA will be complementary to this mRNA, so it will go and bind to the mRNA of an enzyme involved in ethylene production. So when this antisense RNA goes and binds to the mRNA, this mRNA becomes non-functional. the function of this mRNA will be lost. When this mRNA does not function, enzyme activity will be stopped. When enzyme activity is stopped, no ethylene production will occur in the plant. So when ethylene production does not occur, fruits will not get ripened. Means they produce very less ethylene, so they can develop more taste before ripening and shipment to markets. So with the fear of ripening, farmers used to pluck these viruses unripe. These tomatoes when they were still unripe. Green tomatoes were plucked by farmers because if ethylene production starts, then it will cause the fruit to spoil. So when they plucked these tomatoes green, uh, some qualities like taste were still not developed in the tomato fruits. So what they did is they stopped this ethylene production. When ethylene production was stopped, plants, tomato plants, they produced very less ethylene, which caused slow ripening of these tomatoes. So this increased the shelf life of tomato. Such tomato, the transgenic tomatoes which were first produced were known as flavor saver tomato. Now these tomatoes are not in the market. The market now doesn't have this, but these were the first transgenic tomatoes that were produced. They help in delayed ripening, thereby stopping the tomato fruits from spoilage. Okay, this is one of the important trait of transgenic technologies, which was now applied in many plants, in bananas, in potatoes, and in many other soft fruits, strawberries, and all other soft fruits. This technology was applied to delay the fruit ripening by using antisense RNA technology. So here we instead of introducing gene, that is DNA, you are introducing RNA that will go and bind with the actual RNA of the plant. This is the technique here. So the retroviral gene, which it is inserted into the plant cell. This viral genome, which is introduced into the plant cell, it will not allow ethylene to develop in a plant. So this ethylene will not be developed in tomato fruit. When ethylene is not developed, it will not become ripe. It will stay in green color only, and it will accumulate more taste qualities. So when after bringing into the market, if you spray external ethylene, it will ripe, and the plants will be fresh for many days. So how this mechanism is done is, it is produced by blocking the polygalacturonous gene, which is involved in spoilage. So this gene is responsible for spoiling of tomato plant because it breaks down pectin, which is found in plant cell walls. When pectin is present in plant cell walls, which when it breaks down, it will ripe the fruits. So these plants were transformed with antisense polygalacturonous gene, which is mRNA that base pairs with mRNA that the plant produces, essentially blocking the gene from translation. So translation will not happen of the ethylene responsive gene. So ethylene will not be produced. This is the first genetically modified organism to be approved by the FDA in 1994. Then coming to the next quality, that is improved nutritional quality. So more than one third of the world's population relies on rice as a food staple. So rice is an attractive target for enhancement. So because most of the world population, their staple food is rice. If you improve the nutritional quality of rice, then malnutrition or undernutrition will be avoided. In many developing and underdeveloping countries, malnutrition can be avoided if you improve the nutritional quality of staple food, because it is taken in large quantities. So when it is taken in large quantities, more number of nutrients will be supplied to organisms, human body. So in this case, rice was used to improve the nutritional quality, and the transgenic rice which was produced is known as golden rice. This golden rice was genetically engineered to produce high levels of beta carotene, which is a precursor to vitamin A. Vitamin A is required for very many important characteristics in our body, like eyesight can be improved if you have more vitamin A. Low eyesight in individuals is due to deficiency of vitamin A. So for proper eyesight, you need vitamin A. So in this golden rice, you have beta carotene. This is a precursor of vitamin A. Means when it undergoes metabolism in body, This beta carotene will change into vitamin A, and it is supplied to the body to improve the eyesight. So here we use transgenic technology to produce a type of rice that accumulates beta carotene in rice grains. Once inside the body, beta carotene is converted into vitamin A. So this is our normal rice, and this is golden rice, where we have introduced three genes which are responsible for synthesizing this beta carotene. So in this rice also we have beta carotene, but it will be generally present in the husk. So while milling this rice, we will remove the husk. So the essential beta carotene will be lost. So again, for adding this beta carotene, we are using transgenic technology and introducing beta carotene with the help of three genes. That genes are phytoin synthase, phytoin desaturase, and lycopene cyclase. So if you have these three genes inserted into rice, it will help in synthesizing beta carotene. This beta carotene will change into vitamin A during metabolism of rice in our body. So these genes, that is phytoin synthase, phytoin desaturase, and lycopene cyclase, are taken from different other species. Like phytoin synthase was taken from Narcissa, phytoin desaturase from Erwinia, and endosperm specific glutenin from Oryza. These from all these organisms they were taken, and they are introduced into rice for synthesis of this beta carotene. So by this we are improving the nutritional quality of rice. Nutritional quality of rice is getting improved. That will create abolish the or reduce the malnutrition system of underdeveloped countries. So this is also one of the important application of transgenic technologies. Then coming to some other applications, like you can produce tearless onion. Whenever we cut onions, we get tears. So that will be very disturbing. Uh, so by using transgenic technologies, you can create tearless onions also. In fact, the Dr. Colin group, they already created this tearless onions. So the tears, onions produce tears into our eyes because when you slice onions, they release alinases. These alinases are the enzymes which break down sulfoxides to unstable sulfinic acids. When alinases are produced, they break these sulfoxides to unstable sulfinic acid. This sulfinic acid, because they are unstable, they form a volatile gas. This volatile gas diffuses in air, means it spreads in air and thereby reaches our eye. After reaching our eye, it reacts with water to form dilute sulfuric acid. Okay, so this sulfuric acid will help give burning sensation to our eyes. At that time, the tear glands of our eyes, they produce tears to wash off this sulfuric acid. That's why tears are produced while cutting onions. This is because of alinases which produces sulfuric acid. So to wash out this sulfuric acid, tears will be produced in our eyes. So what we will do here using transgenics is we will shut down the lacrimetal factor synthase gene using RNA interference technology. So the conversion of valuable sulfur compounds to tearing agent was inhibited. So in this onion, you use RNA interference technology, means you will interfere with the RNA, same like our antisense RNA technology. RNA interference is also same. You will interfere with RNA synthesis by adding the antisense RNA. So when you stop the RNA synthesis of this alinases, then it will not break down the sulfoxide to the sulfinic acid. Thereby, no tears will come. Okay. So what we are doing here, we are doing gene knockout or silencing, gene silencing by using RNA interference technology. So you will silence the gene which is responsible for converting sulfur compounds to tearing agents. Okay, when you shut down this mechanism, tearless onion will be produced, and it will be very helpful to all uh, in cutting the onions. Then coming to another application, production of purple tomatoes. So what is the need for producing purple tomatoes is it will accumulate anthocyanins. Purple tomatoes, they accumulate anthocyanins which offer protection against many diseases, life risk diseases like cancers, cardiovascular diseases, and age related degenerative diseases. So from Snapdragon, two genes are taken and they are introduced into tomatoes, which generate purple tomatoes. So these purple tomatoes will give many uh, benefits because they contain anthocyanins. Anthocyanins are the pigments which help in combating all these diseases. So this is one of the technology of transgenics, where you use same RNA interference, where you use transgenic technology to generate purple tomatoes. Then with the help of transgenic, you can also enhance the quality. We are enhancing nutrition in golden rice, whereas the quality of fruit can also be enhanced through transgenic technology. Like if you see this apple, whenever you cut an apple, it will turn brown. This is because enzymatic browning occurs in apples or in bananas or in any brinjals and some other fruits. So this when you cut an apple and expose it to air, this becomes brown in color. Why? Because this contain a compound known as polyphenol oxidase. This polyphenol oxidase of one cell mixes with polyphenols of another cells of this apple. When all the polyphenols they mix with each other, uh, they produce a compound known as melanin. Brown toned melanin is produced that will cause the apple to turn brown. This is known as enzymatic browning. So with the help of transgenic technology, you are creating arctic apple variety. This apple variety, even you cut it and keep it for 1 hour, it will not turn brown, because with the help of transgenic technology you are shutting down this enzymatic browning reaction. Enzymatic browning reaction is shut down with the help of transgenic technology. So this arctic apple is a fruit engineered to resist browning after being cut. Okay, these are in use in the US now. So here, what is the main application of this is to cut down on food waste. Whenever an apple turns brown, we will waste it. So in order to avoid that food waste, this quality enhancement was introduced using transgenic technologies. Then the another application is molecular farming, where you produce many important valuable compounds like pharmaceuticals and vaccines using plants, using transgenic technology. So plants may possibly used as bioreactors. So normally in bioreactors which are present in bio industries, they are very expensive, and they uh, in their place plants are getting introduced, and many valuable compound, because plants can be easily grown in farms, whereas for bioreactors you have to maintain industries, you have to maintain very sterile conditions, because any contaminant in that will uh, disturb damage all the product. So there will be very it will be very expensive. So plants, as they can be directly grown in farms, you can get large amount of product using plants. So it is a very valuable application of transgenic technology, that is molecular farming. So here plants offer unique benefits for production of pharmaceutical proteins, like ease of scale up. Easily you can scale them up, means in a field of 1 acre you can grow many plants. Whereas for bioreactors you require large industries with huge bioreactors, huge vessels. That can be avoided here, and they will be low cost when compared to bioreactors. They will be cheap and reduced capital expenditure, and there will be no animal contaminants. In a bioreactor, if any viral or fungal contamination is there, the whole product will go waste. That will be avoided in plants. So this is the one more application of transgenic technology. Then these are the some of the top 10 genetically modified foods. So in market you can see bt corn or bt maize. Then you see soya beans, cotton, bt cotton, rainbow papaya, golden rice, then canola oil, which has improved quality. Nutrition is enhanced in rapeseed oil using transgenic technologies. Then you have transgenic potatoes from tomatoes, you have transgenic dairy products, and transgenic peas. These are the top 10 genetically modified foods. uh, so In these, currently we have BT corn available in the market and BT cotton, and these need permission now. These are the different GMO foods with genetically modified organism foods which are present in the market. So all these, they have different characteristics in them. Like in tomato you can improve the shelf life, in rice you can improve the nutritional quality by adding beta carotene and vitamin A. In maize you can make it insect resistant, so any caterpillar may not attack, cannot attack it. Like that, in all these GMO foods you have certain important characteristics which will improve its production. So these are some of the benefits and risk of GMO. So what are the benefits of genetically modified organisms are: the nutritional value can be improved by introducing proteins, vitamins and vaccines. Then they can be produced that lack known allergens. Many people, they will be allergic to many plants, like peanut allergy will be there to many individuals. That in that peanuts, whatever the allergens responsible for that allergy can be removed using transgenic technology. Then crops can be grown in arid conditions, like in dry conditions also. If you introduce any drought resistant gene or saline resistant gene or flood resistant gene, you can grow that plants even in floods or mangroves or in drought affected areas also. So they give better yield even though there are arid regions. In dry regions also, if you have drought resistant plants, they will give better yield and productivity. Then GM crops can produce herbicides to kill pests, like we have seen herbicide resistant plants. Then this is the most important one, that is they improve the food supply in poor countries. You can use foods which give higher yields. Then they have longer shelf lives, so less spoilage and reduced economic cost and carbon footprint. So when your carbon footprint is mainly responsible for this global warming, so when you introduce these GMOs, carbon footprint will be reduced because you use less land and less pesticide usage. Because when they are pest resistant and insect resistant, there will be no need of using the pesticides. So you are protecting the earth from global warming. Some of the risks are: new traits can cause adverse health reactions, very minute, but sometimes the new characters which are introduced into plants, they cause adverse health reactions like allergies and others. Then removal of traits could have unknown effects. If you remove some unknown some traits or characters, they have unknown effects. For example, here we can say that when you introduce herbicide resistance into plants, the weeds will get killed due to herbicide spray. Like we have discussed about glyphosate. When you are using glyphosate or Roundup Ready spray, that is damaging, destroying all the weeds. But when these weeds are destroyed, some of the insects in the ecosystem, they will feed on these weeds. So automatically ecosystem will get damaged. That is, the insects which feed on the weeds, they will also not survive because there are no weeds. You are spraying the herbicide and you are removing all the weeds. For example, monarch butterfly, the butterfly, yellow, orange and black color butterfly which we generally see, it is known as monarch butterfly. It will feed on milkweeds. Milkweed, it is a weed which grows in between soybean plants or maize plants or cotton plants. So when you are spraying herbicide Roundup Ready, these milkweeds are getting dying. It leads to death. So when you are removing the milkweeds, this monarch butterfly larva, it will monarch butterfly, it will lay eggs on these milkweeds. So when milkweed is not there, then your monarch butterfly has no place to lay eggs, and automatically it will also gets removed from the ecosystem. That is one of the risk of these GMOs. Then you have patents. These patents, they will restrict farmers to easily use these GM crops, because when you issue patent, when you issue patent, it will be in the hands of only some. It will not be freely available in the market, so you will not use it. Farmers are not free to use them. Okay, these are then there will be many government regulations and controls to use these GMOs. One of the disadvantage of these GMOs is terminator genes. So in this, uh, farmers have to buy seeds every year. They cannot save their seeds from this their crop of the current year. They cannot save the seed, but instead they have to buy fresh seeds every time from the market, because the crop, the GMO crop which is introduced, which they use like BT cotton or BT corn, which they have used, they contain terminator genes. These terminator genes, they will make the seeds sterile. When when these seeds become so these GMOs, they have terminator genes which make these seeds sterile. So the seeds which they have used to grow their crop now, the crop which again gives the seeds, they cannot save them for the next season. Instead they have to for every season they have to buy fresh seeds, because these GMOs, they have a technology known as terminator technology, where there will be three genes. These three genes will not allow the seeds to become mature, means mature seeds will not become fertile, they will be sterile. So this is the technology used by genetic the manufacturers of genetically modified organisms, which will not allow the farmers to save the seed and use for next season. So this is the history. In the first transgenic plant was produced in 1985. Then in 1988, the direct method of gene introduction, that is gene gun method, was introduced. Then in 1994, flavor saver tomato was released. In then in 2000, golden rice was released, and now if you see in the market, we have many GM crops, but still the debate is going on whether they are beneficial or risky for human health. So this debate will always go on, and these are the some of the leading agribiotic companies like Monsanto and Syngenta. These two are the leading companies which provide the genetically modified crops, seeds for growing BT crops. So there are certain concerns regarding transgenic plants, that is cultural or religious issues, like many times genes from animals were introduced in plants. That is one of the concern of transgenic plant. Then dietary concern, because there may be some allergies for by when you introduce some novel proteins into plants, they may cause allergies. Then there is gene escape, means the weeds which are now herbicide sensitive, they may become tolerant in due course of time. They may become antibiotic resistant or antibiotic tolerant. So because gene may escape from roundup ready beets or roundup ready soybeans into the weeds, the gene escape may happen. Then wasting precious genes one at a time. That is, widespread use of single Bt genes could provide intense selection pressure for resistant insect, rendering the use of Bt spray ineffective. So when you are using Bt genes for, so you will, there will be an intense selection for resistant insects. Then there will be a conflict for ownership, because the genetically modified crops are always subjected to patents. Natural varieties, they are non-patentable, means there will be no right, anyone can freely use them. But these GMOs, only some people have the right to use them. That is to use them, to sell them, to make copies, to make license. Only some people will have the right to use them to make benefit. So that is known as patent. So that will be a concern, because no one anyone is not free to use any of the GM crop. They have certain, they have to take certain permissions to use GM crops. So these are some of the concerns regarding transgenic plants. Then what are the future directions is, you can GM crops, genetically modified or transgenic crops, they serve as valuable alternative in solving food security problem. Definitely, because uh, these transgenic crops, definitely they will help in food security. Means we cannot make our future generations to starve. So we can use GM crops instead to make their future secure. However, transgenicity remains a major controversy in view of biosafety issues. So there are many misconceptions in public related to GM crops, but uh, if we do further research work and if we do risk assessment, we can definitely remove these misconceptions from the public. Like when a bt cotton or bt brinjal is affecting insect gut, it may also affect human gut. That is a misconception. By doing further research and further scientific metabolic and molecular research, you can remove these misconceptions and grow GM crops in the same condition as non-GM crops. So the conclusion is the ultimate goal of transgenics, that is introduction, integration, and expression of foreign gene, is to improve the crops with desired traits. So the goal of transgenics, ultimate goal is to improve the crops, improve the plants and crops by introducing desired traits. Like in transgenics, you are introducing a gene, you are integrating it with the plant genome, and you are expressing that gene to improve the traits. This is the ultimate goal of transgenics, and I think we have succeeded in this goal of transgenics, and now we have many GM crops. We have many GM crops that are helping us to improve our food security. So with this, I would like to end my presentation. Thank you all.
Thank you, ma'am.
Any
Wonderful
questions?
presentation. So many concepts you have
So,
dealt. Really, it looks very fine that browning of apple. So many concepts you have told. Really, it was a bundle of information you have given in a short span. Great knowledge you have shared today. Really, such kind of more meetings are required from our neighboring departments so that everybody can understand what is going on and what is to be done and how we have to change as we have come in the age of I mean, it's a mobile age, or we can say the age of Android age. So many things are required. We need to update ourselves. Thank you, Javeria Uzma ma'am, for such a wonderful presentation. Um, have a nice day. You have given a really wonderful lecture, and I will be requesting you in future also for such many more lectures. Thank you. And now I request Anjana ma'am to start sharing her screen. Ma'am?
Yes, good
Yeah.
afternoon to all.
Yeah, good afternoon. Yeah, our next speaker is from Kathmandu, Nepal, Tribhuvan Das University, Professor Anjana Singh, ma'am. Ma'am, give me a few, some time. I will introduce yourself and then you can start your presentation. So, Anjana Singh, ma'am, is academician, Nepal Academy of Science and Technology, Nepal since 2010, executive member of Special Committee of AASSA, Women in Science and Engineering since 2013. Former doctor, former head Central Department of Microbiology, Tribhuvan University, Kirtipur, Kathmandu, Nepal. So her education is from Kathmandu, Tribhuvan University, Kathmandu, Nepal. She has done her MSc, BSc, both from the same university, and her PhD is in microbiology from School of Life Science, Jawaharlal Nehru University, New Delhi. Her postdoc was from University of Virginia, USA. Her major activities were from 2004 to 2008 and 2012 to 2016 were head Central Department of Microbiology, Tribhuvan University, Kathmandu, Nepal. She was executive member of Board of Studies, South Asian University, New Delhi. She was executive member of Nepali Society of Microbiology, President Filters for Families Nepal, Associate Academician of Nepal Academy of Science and Technology, academician, executive member of Fulbright Alumni Association Nepal, Women in Science and Technology Nepal, Health executive member, Health and Environmental Welfare Association Nepal, life member of Nepal Biotechnology Society, UGC Cluster Committee Board Member, TU Research Committee Member, and Subject Committee Member for IOST Tribhuvan University, BOS Member of South Asian University, New Delhi, NAST Senate Member, NAST Research Committee Member, Board Member, NAST Management Committee Board Member, Organization for Women in Science in Developing World, Coordinator of Women Scientists Forum Nepal. Honors and awards to her credit are Fulbright Scholarship, United States Government, Education Award, Shiksha Puraskar Nepal, International Foundation for Science Grant Sweden, Crown Prince Young Scientist Award Nepal, Mahendra Vidya Bhushan Award Nepal, University Grant Commission Fellowship Government of India for PhD, University Grant Commission Mini Research Project Nepal, Tika Lakshmi Padak Tribhuvan University Medal for securing highest mark for among girl students in BSc Nepal, Mahendra Vidya Bhushan Award in BSc for topping in BSc 1991, Birendra Aishwarya Scholarship 1991, Aishwarya Vidya Padak for standing top among girl students in Nepal, Harihar Raj Lohani Award for standing top taking chemistry in BSc Nepal. Her publications in national and international are 125 and peer-reviewed journal 60, books 3, book chapters 25, general proceedings publication 28, abstracts published 9, accepted articles on pipeline 9, supervision 1999 till date.
What happened?
Supervised 103.
Yes, sir.
Guidance Supervisor 1, Co-Guide Immunology and Infectious Diseases Plant Health. With these words of introduction, I would like to request kindly ma'am to start her presentation.
Good afternoon. First of all, I would like to thank Professor Khan for such a nice introduction of mine. And I'm also very much thankful to the organizers for the invitation on this lecture series, Department of Botany, Telangana University. And also, obviously, I'm very much thankful for Professor Khan for reaching out me. The topic of my presentation today for the students is Fruit Tonic for plants. My slides are not moving. Okay. So when we talk of Fruit Tonic, I just wanted to have a little bit of catchy topic for the students, because most of the time roots of plants, they have various symbiotic effect of bacteria and fungi and etc. for its growth promoting effects. So here I'll be mostly talking with the of mycorrhiza. So what is mycorrhiza, right? So when we talk of mycorrhiza, it's a word that has been coined by Frank, and it is the effect of this mycorrhiza with the plants is mutual symbiotic association between a fungus and a plant, and it refers to the role of the fungus in the plant's rhizosphere. We know what is rhizosphere because there's a rooting system, and the rooting system surrounding the roots are known as rhizosphere, and on the plane of the roots surface, it is known as rhizoplane. So there are various types of bacteria, various types of fungi. It inhabit around that surroundings, and it also helps the plants for its growth promotional effect. It's not only that, sometimes there are pathogenic organisms also which might harm the roots or the plant. But here we are mostly taking in the positive sense. And mycorrhizae, we say in plural, that is the mycorrhizae, and for singular we say mycorrhiza, play important roles in plant nutrition, soil biology, and soil chemistry, that is the whole soil biota. Here I have shown the picture where there is to the left with mycorrhizal fungi, and to the right is without mycorrhizal fungi. These are the rice seedlings, where you can see that there is a whitish, lots of hairy roots coming up in the rooting system. You can see here below, these are all mycorrhizal fungi associated with the roots. And to the right, that is without mycorrhizal fungi, means it's a control experiment where you can see only the plants or plantlets coming up with its roots only, devoid of mycorrhizae. And this mycorrhiza, this word has been coined by A.B. Frank in 1880, and it has various types of mycorrhiza, where one is ectomycorrhiza and another is endomycorrhizae. So when you say ectomycorrhiza, there is mycorrhiza, or you say that it is a fungus which do not penetrate the individual cells within the root. But just opposite to this, endomycorrhiza, it has association with the fungal hyphae, and it is present on the root surface, and the threads of that rooting system or the mycorrhizae, it penetrates directly into the root hairs and epidermal cells and into the cortical cells of the root. Here in this picture also you can see that endomycorrhizae penetration, where it has entered into the split-like cells of the cortex region of the plant, and you can see that there are lots of arbuscles vessels has been developed. Similarly, here I have actually this picture. I'm very sorry for this. The picture has been exchanged. The earlier one was with the endomycorrhizae, and this one is for the ectomycorrhizae. Here you can see that in the ectomycorrhizae, the difference between endo and ecto is that in the ecto, the fungus, it grows outside the root surface. Okay, but it does not penetrate within the cortical region of the root cell. But in the endomycorrhiza, the name itself, endo means inside, it penetrates the cortical region and it develops the arbuscles within the root cells. And the difference here is in ectomycorrhiza is at the outside of this root, you can see that there is a network. You can see very hairy type of network, which is known as Hartig's net also. And it is present between the cells. And another word is there. One is ectomycorrhiza, one is endo. Ecto means outside, endo means inside, and ectendomycorrhiza, it's a combination. The fungus belong to Basidiomycotina, which covers both gymnosperms and angiosperms of the plants. These mycorrhiza, it show many of the same characteristics of endomycorrhizae, but also show extensive intracellular penetration. So, the formation of ectendomycorrhizae begins with the formation of this Hartig's net, similar to ectomycorrhiza. Then it grows behind the apical meristem of the growing root, and this Hartig net, after that, it's a network outside the root surface. So after this Hartig net formation, these will start to penetrate between the epidermal and outer cortical cells, and later it extends to the inner cortical cells of the roots. Now, what are the applications of mycorrhizae? When you talk of mycorrhizae, these increase nutrient uptake of plants from soil, like phosphorus nutrition and other elements, nitrogen, potassium, calcium, magnesium, zinc, copper, and so on. And then there are so many things, so many these minerals that are very much important for the growth of the plant and its proliferation of the plant. Sometimes it is very much important in the growth, or sometimes it is very much important in the fruiting time or in the seedling time, right? So these increase diversity of the plants among various types of plants. So it produces uniform seedling and significant role in nutrient recycling, more tolerant to adverse soil chemical constraints, which limit crop production. Because after some time, what happens in the soil where the nutrition is there, it gets exhausted, right? But with the help of these mycorrhizae, it goes far away from the roots and it collects not only the nutrients, also the moisture from the soil, and it will transfer to the root of the plant. So this will, the rooting of the plant, it will help to absorb the nutrition and the moisture to the plants, to the various parts of the plant, and increase plant resistant to various diseases and drought also. Previously, Madam also talked about the drought condition, how genetic makeup has been done and produced, right? But here, these mycorrhizae also help in stopping various type of pathogenic microorganisms entering to the system in the plants. Not only that, sometimes there are some adverse condition of the environment, that sometimes there are lots of water is present, sometimes drought, there is no water available, sometimes the pH of the soil is different and is very harsh, and sometimes salinity is very high where most of the normal plants does not grow. So these mycorrhizae, it will help the plant to grow in or survive in that adverse condition. And it will stimulate the growth of the beneficial microorganisms. Like as I have already told you, that besides mycorrhizae, there are other fungi also, which is very much important for the plant's growth. And there are other bacteria also, which is very much important, like you have been studying from bachelor's level also, that there are some kind of PGPRs, right? Plant growth promoting rhizobacteria. These bacteria also, it will stimulate these bacteria to survive in that condition and help the plant for the plant growth promotion. And it will also condition the soil. That means it will improve the soil structure. Therefore, this plants can survive easily and grow easily. So these indirectly, it will also help to stable soil aggregation. If the soils are aggregated to each other, then this soil will not flood away when there is lots of rain is there, or these nutrients which has been trapped within the soils will be easily absorbed by the rooting system of the plants. And as you know, that hyphal condition, it will help with the help of polysaccharides that might bind and aggregate the soil particles to each other. And it will increase absorption of phosphate by crops. Like why I'm saying phosphorylation is that sometimes the direct added phosphate can be easily absorbed by the crops, but sometimes rock phosphates, which is present in the nature, is unavailable for the plants. So in this condition, what happens? There are some other bacteria also it will utilize, obviously, but here, since I'm talking of mycorrhizae, these mycorrhiza, it will help to degrade this rock phosphates into mineral phosphates, and it will be easily absorbed by the rooting system of the crops. And it will uptake zinc and also increases in the conditioning or the life of plant. It increases uptake of water from the soil, which I was talking as moisture, and increases uptake of sulfur from the soil. Concentration of cytokinins and chloroplasts in the plants will be increased. They will protect during the stress condition. So this is the line drawing I'm trying to show where the various types of mycorrhizal fungi is present, because at one go it is very difficult to show that how mycorrhization has been worked. But here there are 7 types of mycorrhization or mycorrhizal fungi. Like one is ectomycorrhiza, which I have talked about it, and arbuscular mycorrhiza, ericoid mycorrhiza, arbutoid mycorrhiza, monotropoid mycorrhiza, ectoendomycorrhiza, or you can also say ectendomycorrhiza, orchidaceae mycorrhiza. So orchidaceae mycorrhiza, you know very well that orchids, it doesn't have any chlorophylls in its sorry, not chlorophyll. It doesn't have its own mechanism that it can absorb the nutrients material from the soil or from the parasitic host that is being used, right? So what happens? Mostly the mycorrhiza are there for the mycorrhization is available within the orchidaceae plants. So because of that, what happens? Mycorrhiza, it helps to absorb the moisture and the nutrients to the orchidaceae plants. So because of this, the orchids is totally dependent on mycorrhizae. If you do not have any mycorrhizae in the soil, if you just try in the laboratory that if you make the soil sterile and you try to plant orchids itself without any mycorrhization, you'll notice that it won't grow. Otherwise, artificial hormonization has also to be added with the plants. So these are the line drawing shows that how the mycorrhiza, these 7 types of mycorrhiza, is being distributed within the root system. So the mycorrhiza role is overcoming drought and stress condition. So because of this, 90% of plants are colonized by mycorrhiza and mostly economically important plants. Here when we say it is economically important plants, these can be the day-to-day, the food we take is like cereals and crops, and also even we can see the some of the flowers, sorry, floriculture, horticulture, viticulture, these all sometimes they need mycorrhization. So because of that, what happens? The sugars are available for the plants, and proteins like dehydrins and heat shock proteins, these will be also available. And otherwise, these won't be available to the plants, but because of the mycorrhiza, these help to make the plants available. And there are some free radicals also that they will also help in scavenging and osmoregulation of the water uptake for the plants. When you talk of benefits of mycorrhization, obviously the plants are benefited. Connection between root and soil, so because of that we say symbiotic also. And it acts as a growth promoter, nutrient uptake, increase the surface area. That means the rooting system of plant is to the some stipulated area, but this mycorrhization, it will spread along the soil in the ground, and it can even go up to something like 30 feet or 40 feet away. Sorry, I'm having a cracking voice, I'm very sorry. And because of that, these hyphae or the mycorrhizal hyphae that will take the hypha, these will take all the nutrients and water to the root system of the plant, and obviously the plant can survive, and it can also fight against the pathogenic organisms also. And this is how the surface area has been increased. So because of this, it will increase the mobilizing of the unavailable nutrients, like I have already given the example, rock phosphates and its utilization, and also excreting chelating compounds or ectoenzymes. Like sometimes the enzymes has been produced, it can be some organisms, some other microorganisms like bacteria and other fungi. They may have this ectoenzymes in the area. But in the rhizosphere, if it is present, then the mycorrhiza will absorb and it will transfer to the plant's roots. So here I'm showing some of the pictures here. There are some plant transfer metabolites are also there, which will also benefit the plants for its growth promoting effect. And here plants transfer almost 10 to 20% carbon to the fungi also. So this is how we say it is a symbiotic relationship between the fungi and the root system or the plants. I have taken one example, Piriforma spora indica here, which is a part of my PhD work Nowadays, this Piriforma spora indica is also known as Serendipita indica. So this is the same organism. Since the time passed away, the name of the organism is changed, and we still call Piriforma spora indica, but the Western world they call Serendipita. So here, some of the spores of the mycorrhiza I have shown here. These are the endomycorrhizas. So after sporulation, it can be observed in the soil, where you can also get these soil spores from the soil after a few processes, where there is washing the soil in various ways and then sieving it, and there is a long process for this. But however, it's not that difficult. You can observe or you can obtain these spores. So these spores can be also directly added to the plant seedlings. And here to the right, you can see is this Piriforma spora indica. These brick-like structures are the root sections, and lactophenol blue staining has been done, and these are the spores of that Piriforma spora, where the name got that is it has a pear shape, thus it is known as Pirispora indica. You might be very inquisitive, or you want to know that this Piriforma spora indica was accidentally isolated with association of spore of Glomosmosi. Here, Glomosmosi, this is the Glomosmosi spore, where you can see one of the PhD student was working with this, and she was isolating this Glomosmosi, and it's very surprising that there was one student who was quite active, and the guide told the student, try to culture this spore on a synthetic media, that is the nutrition media, MN media, and every literature says that Glomosmosi cannot be cultured in the media. But however, this PhD student was able to grow in the culture media, that is synthetic media in the laboratory, like you people also do culturing in the laboratory, right? Similarly, this student was also doing it. And again, she saw the culture growing. And she told the supervisor that this is growing. But how can it be? The supervisor was not believing, because everybody in the world knows that Glomosmosi cannot survive on the synthetic media, because it needs plants' rooting system for the growth, right? So again, supervisor told her to do the growing of that Glomosmosi on the synthetic media again and again. And because of this, what is happening? The student was growing this for so many times and so many times that almost for 6 times she grew this thing on a synthetic media. And because of this, what is happening? Supervisor not believing. And later, when supervisor then sat together and then saw this culture media, in the culture media, that the student was thinking that Glomosmosi was growing, but later it was found that it was something else that has been growing. That the soil was obtained from the rhizosphere of the 2 shrubs in the Indian Thar Desert, northwestern Rajasthan in 1990, and here the paper is I have shown is Virma et al. In 1998. The paper was published, and this she is the student, and supervisor is Verma. This in 1999. Here you can see where I'm showing the arrow is Verma, VAR. VARMA is the supervisor, and VERMA is the student. So this is how accidentally the fungus was found, later named as Piriforma spora indica. So with this message, I would like to give the students also that when you do the research work, when you do your work in the laboratory, don't get disheartened, right? So you never know that you will be finding out, or you are maybe you can also get some kind of new findings in your experiment. So there are other arbuscular mycorrhiza like fungus are there, and this fungus is known as AMF-like fungi. We don't say arbuscular mycorrhizae, like you say for the Glomus mosaic and rest of the other fungi, endomycorrhizal fungi, because this is mimicking all the characteristics of Glomus mosaic and other endomycorrhizae fungi. But the difference is they are unculturable in the synthetic media. They can grow within the rooting system of the plant, but they cannot grow in a synthetic media or in a plate in a laboratory. However, this Piriforma spora indica, it is growing. It can be exogenically cultivable on synthetic media. It doesn't have any clamp connections. Anastomosis occurs frequently. Hypha-hypha aggregation is often observed, and no hyphal knots is seen. Simple septum with dolichopores and continuous straight parathizomes are being seen, and chlamydospores were about 16 to 25 micrometer in length and 10 to 17 micrometer in width. So this is a little bit smaller than compared to this Glomus mosaic. So it has about 8 to 25 nuclei per square spore. Because I will go back to this slide again to show you. Here, to the extreme right down, see, these are the nucleus that you can see. There are so many, almost 1 to 25 are present. So this is, I had been lucky to find out this staining, trypan simple staining in the root system. This is inoculated with the Piriforma spora indica. Problem in moving my slides. Okay, the functional similarities with AM fungi are the following. It has broad and diverse host spectrum as other endomycorrhiza. Hyphae extramatricial inter and intracellular can be seen. Hyphae never invade the endodermis, and chlamydospores in the soil and within the cortical tissues are being seen. Sexual stages never seen. Positive phytopromotional effects on tested hosts, and phosphorus mobilizer, phosphorus transporter, tool for biological hardening of micropropagated plantlets, and potent biological control against various root pathogens. So it has been tested with various pathogenic fungi also, that like wilting and all these happens with the fungi. So on these fungi also it has been tested, and it shows that this Piriforma spora indica, it inhibits the growth of this fungi. And biological hardening here we say is that when the plantlets are ready to be transferred to the field or ex situ condition, these have been incorporated with this fungus or this Piriforma spora indica. So this is known as biological hardening in the micropropagated plantlets. So it can survive on the harsh conditioning effect on the ex situ condition, and also promote the phytopromotional effect of the plants. So there is one experiment that has been done also in Piriforma spora indica protoplasts. It has been applied PCR with specific primers. It indicated that a similar kind of fungus apparently occurs in the rhizosphere of Leptocloa fusca or Oryza minuta in Pakistan. So when this was identified in India, similarly, same type of was also identified in Pakistan and Philippines, but however, they are not exactly the same. And the whole spectrum of Piriforma spora indica is very much like AMF fungi, where it can be calculated that AMF fungi interact with almost 90% of the terrestrial plants. And this also colonizes the roots of host plants as diverse as economically important plants. Not only that, various crops, medicinal plants, and all, even the forest, the tree seedlings Also, it has been tested and it can also colonize these, and obviously it can be also planted in the forests with along this fungus. One of the striking differences is that unlike AMF fungi, the host range of P. indica also includes terrestrial orchids. Earlier also I have shown you that, already told you that orchids are also incorporated with this P. indica, and it gives a very good proliferation of the orchid plants. And here the scientists from MIT University, they have coined this the word rootonic, because this Piriforma spora indica, it had been studied almost more than 150 plants, and it has a promising output of laboratory experiments, and they have also studied in small field trials, indicating the need for its mass cultivation and usage. So for field trials, a formulation rootonic from certain secondary metabolites from the fungus was prepared, and it is mixed with Piriforma spora indica biomass in magnesium sulfite. So they have given the name as a rootonic, and this has been sprayed, and or it can be applied, or sometimes the seedlings can be dipped in this solution and then planted, right, as a biological hardening for the micropropagation of the plantlets. And it has a standardized for almost 50 plants, and Piriforma spora indica has proved to be a very highly beneficial endophyte with highly efficient in field. So they have reviewed an article that on their journey with the Piriforma spora indica to rootonic. So that is how this term has come up here as rootonic, and it was published in African Journal by Srivastava and Verma in 2021. And here to the left, I'm showing is this is an electron microscopy of Piriforma spora indica. Since it is a pear-shaped, it is known as Piriforma spora indica because from India it has been found. And the hyphal coil is often seen. See how nicely the coil hyphae can be seen. And there are chlamydospores, that these are the chlamydospores obviously. And when you magnify this hyphae, you can see that there is presence of dolichopores and aprosoria. See? And this is I have taken from the net that they have from the MIT University, the scientists, they have worked on it, and they have given the name rootonic for this fungal mixture, where they are applying for various fields in case of promoting agriculture, horticulture, and forest productivity. And I would like to give you how this mycorrhization can be prepared for using in the plant seedlings. The first method, here you have taken the soil inoculum in this Piriforma spora indica with the soil. Obviously, it is in a sterile soil that has been produced. And this inoculum that you first take the substratum, that is the soil sand composition. three parts of soil and 1 part of sand has been taken. For soil, you have to do sterilization, otherwise you won't have only the organism that you are going to add in it, right? Or that you are going to add as an inoculum. You'll get lots of from the environment or in the nature, what has been present. So to kill all these spores, first you take the soil and autoclave as in usual way for 3 successive times. Means one time you autoclave, suppose if you autoclave today for a whole night and whole day, next day you leave in the environment. Environment means not open, like you put in a plastic, the heat resistant plastic, and do autoclaving, then put it for some time, whole day and night. Next day again, 1 day after, then again you do the autoclaving. And again, you take it out from the autoclave and keep it in the cool place. And don't open it. If you open it, you know that you are going to again add the microorganisms from the air also, like from the environment. So don't open it. Then again, cool it for whole night and day. And again, after next day, you do the autoclaving. That is 3 successive autoclaving you do. That is how you will going to kill all the spores or microorganisms present in the soil. And for the sand particles, what you do, you take the sand, wash thoroughly in the tap water, and those which has been clean sand, you put 1 normal HCl for whole night. Put that sand for the whole night in the 1 normal HCl. Then after then, the next day, slowly you decant that HCl, but be very careful since it is acid. To wash all the traces of HCl present, and you do the testing of that pH, whether that the soil, sand, water has come into a normal or not, right? After that, then you dry both of the sand and soil, then mix in 3:1 composition, and that is known as substratum. So you take this as a plantation of the seedling. Now this is the Zea maize seedling that I have shown in the figure. So after some time, 2 to 3 months, it will take time that there is a sporulation of that inoculum, and it will penetrate the rooting system of the plants, and that is how you get the inoculum in mass production also. The same process, another way of mycorrhization is that we have developed this in the laboratory. You take the inoculum, that is the Petri plate. I have told you that though it is an endomycorrhiza, it is culturable in the synthetic media, right? So this has been grown in a NMM media. So in the media, you grow it, like it takes about 5 to 7 days to grow fully in the plate like other fungus. Then you cut, or you take a clean, sterile cork borer and then cut the agar containing this inoculum in a various sizes in a disc form, and these again in the earthenware pot you put the substratum like as I have shown you in the figure, and in the dip in the middle you just take a wooden stick, clean grease-free clean wooden stick, and then poke. So that is the way you make a hole in it, and in that hole you make 1 or 2 discs in the as an inoculum. Then again you cover with the substratum after the planting of the plantation. This is how the mycorrhization can be done in the laboratory condition, and also from greenhouse to the ex situ condition. Next. There is axenic root culture of this arbuscular mycorrhizal fungi. These are the glomosmosi, which I am showing. These are grown in the, it can be, as I have already told you, similar process that I have shown you just 2 slides before. These are, I have shown you with the Piriforma spora indica, but same process can be used with the glomosmosi and other endomycorrhizal fungi also. But these, now right now in this present slide I have shown it is in the root organ culture. Here carrot is sliced in a small pieces, and in this sterile carrot pieces, it has been grown in an synthetic media in the laboratory. That means it is not in the soil, right? So here you can see lots of sporulations, and for your experiments you can get lots. Now, when we say biological hardening, here Botany somnifera, it is a medicinal plant. You all know that. And here we have shown here that with mycorrhizal and non-mycorrhizal, MIC is negative, that is non-mycorrhizal, and MIC positive is with mycorrhizal. You can see the differences here. It has a very good flourished. You can see lots of number of leaves are also increased, and the plant is also healthy. While in the non-mycorrhizal plant, you can see there is disease, a little bit disease leaves have been seen. So with this also you can see that the plants can be produced as a disease-free plants also. So in another condition, that Piriforma spora indica, when we talk of antigen antibody complex or immunoassays or immunology, we always think of having the disease that is related with human or with the animals. But it's not only that, it's not the immunological aspects cannot be used only for the disease in the human or animals, but it can be also used for the identification or characterization of the fungi. Here, fungi, the Piriforma spora indica, is being used for the identification of its whole. Obviously, the whole genome has been identified by somebody else in later, but this is a portion of my experiment also, that the spores of Piriforma spora indica has been isolated in a broth culture, and it is concentrated, and it has been crushed to isolate the proteins of it, and it has been injected in rabbit, the New Zealand white rabbits. And after the successive inoculation of the spores in the rabbit, what is happening? The rabbit is making antibodies against these antigens, right? So with this antigens, the rabbit produce the antibodies. Now I can easily identify the antigens of various, from the various soils, I can easily identify the presence of Piriforma spora indica. So Piriforma spora indica, the antigen itself is being isolated from the broth culture, right? So that I take in a various, what you say, in a ELISA plate, and the antibodies that has been raised indica in the soil or not. So same thing, but it is used for the dot plot assay procedure for identification of Piriforma spora indica. Dr. Khan, am I audible? Dr. Khan, am I audible? Dr. Khan, am I audible? Dr. Ahmed Abdul Halim Khan, can you hear me? Because it seems that my network is not working properly. Anyway, and there are some of the mycorrhizae, but these are ectomycorrhizae, which can be also used. But the Piriforma, to the left is the Piriforma spora indica, and to the right is Sebacina vermifera sensu. So both of these fungi, it mimics each other, and they are almost similar, but however the little bit difference in genetic composition. And this Piriforma spora indica, it is found in the found from the soil of Indian soil, but this Sebacina was found in Germany. So it has almost the similar morphological characteristics. And this is the ectomycorrhiza. As I have told you, endomycorrhiza cannot be grown on the synthetic MMN media, but ectomycorrhiza can be easily grown on the MMN media, where I have given almost this 12 ectomycorrhiza, which has been grown on the MMN media, just to show to the students. And this is why we are saying Piriforma spora indica. It is AMF-like fungi, because it can be grown on this media, and it mimics the characteristics of all the endomycorrhiza. And when mycorrhiza being used, here we say it is the Piriformaspora indica. This is the control. A is the control with the Zia maize only, without any mycorrhization. And this in the middle, B is the Piriformaspora indica being added, and to C is the Sebacina vermifera. So both of the mycorrhiza, it's showing good proliferation of the plants. And similarly, in the rooting system also, when we say, when we have taken this rooting system of the maize plants, you can see that in the control plant, the rooting system, it is very poor. With P. indica, it is very nice. And similarly with vermifera, Sebacina vermifera, which is also AMF-like fungi incorporated. And the last one is the Glomus mosi, that is the endomycorrhiza. Here, the for the identification of Piriformaspora indica and vermifera, it has been used in the various broth cultures and its growth comparison, which is almost coming similar. And also in the cytochemical staining, here you can see with the trypan blue, where it is also coming similar. And here I can show you is that this Piriformaspora indica, and it is this in the middle, it's showing in the trypan blue staining where the sporulation is being seen here. It is a plant promoter, biofertilizer, acts as a biofertilizer, acts as a bioregulator, also acts as a bioherbicide, and
Ma'am, you have to unmute yourself. You have gone mute. Ma'am, unmute yourself, ma'am. Yeah.
host as muted has come up. Okay, let it be. I'm almost done. And it also works as a phytoremediator, biocontrol against insects and pathogens, stimulator with PGPRs, antioxidants and drug enhancer, enhances anti-aging agents, this has been recently done, and promotes anti-cancer drugs and many more to come. The source as I have taken from the ResearchGate, Amit Kharkwal, who is also working with my mentor, PhD mentor, Professor Dr. Ajit Verma from MET University. With this, I would like to thank again for listening to my talk. Thank you.
Thank you very much, ma'am. Such a wonderful presentation of yours. As usual, you are the person who did a great work, a job for improving the agriculture. So this is the work which is need to be made more and more popular among the people, so that it can bring the sustainable way of cultivation and sustainable practices. So with these words, I'd like to once again express my deep sense of gratitude towards the speakers of the day, as well as the participants. Kindly participants, note that we have 2 more lectures is scheduled. Tomorrow we have 2 more speakers, and Monday also we have 2 more 3 lectures. So after that, we'll be sharing the feedback for certificate. Thank you very much for all the participants. And here we end the meeting for on and all. Thank you, ma'am.